So what about today? Where are we and what's really around the next bend? Well, first of all, I'm just going to summarize where we are with our present picture of prion disease. We have a gene, we all have it, and it's translated to make the normal prion protein, which we're all making now, all of us. But in disease, this converts to the abnormal form, PRPSC, and then there's this amplification process that occurs whereby the abnormal form turns the normal into more abnormal, and the abnormal protein gets deposited in tissues. But why people become ill is because brain cells, neurons, become ill, malfunction, and die. And that's a very important part of the process to which I'll return later on. So there's some further detail that you need to add in. It's a bit annoying because the scheme itself was simple enough. The first is that there's a lot of intermediate forms between the so-called straightforward normal form and the stuff that's deposited in tissues. And there's a lot of questions around which forms are relevant for transmission, what are the infective forms, and which forms are responsible for disease. And it's not necessarily the case that this orange blob at the final end that gets laid down in tissues is responsible for either of those. The second important detail is that you see different abnormal protein structures in different prion diseases. So the abnormal form from one disease is not necessarily the same as from another. Well, that's all very well, but what actually sets it off? I've said this happens. Well, there are three causes that make this initial conversion happen: genetic, acquired, and idiopathic. And in genetic forms, the gene is slightly abnormal in some sort of way, and therefore it produces a slightly abnormal protein, and that slightly abnormal protein seems to be less stable and more easy to go into an abnormal form. If it's acquired, basically this abnormal protein from an external source enters the body and forms the first conversion step. And acquired disease is Kuru in the Eastern Highlands of Papua New Guinea, variant CJD related to BSE and food, and iatrogenic CJD related to certain medical and surgical treatments, but particularly in the past, human growth hormone and durameter derived from cadavers and used in surgery and medical treatment. Then we come to the fact that actually most of the disease is what we call idiopathic, i.e. of unknown cause, or sporadic, because it just occurs sporadically throughout the world, everywhere where it's looked for no good reason. And the present theory is that this is just some sort of spontaneous error. We're making these proteins all the time, millions and millions of molecules throughout our life, and something somewhere happens to go wrong, a spontaneous production error. So if I talk about sporadic prion disease, it's the commonest form of human prion disease, over 80% of it. It leads to about 1 to 2 deaths per million per year. That's what we call the annual mortality rate. But the lifetime risk of an individual developing this disease is about 1 in 5,000. It's thought to be spontaneous, and all I can say is there's been a lot of research over many years into many other theories, all of which have proved negative. Interestingly, it's not a uniform disease. There are variations in the clinical and pathological picture. So if there are variations, that does raise the question of, well, what governs the way a prion disease affects someone? Well, first of all, the gene. It's not only that the gene has mutations which cause genetic disease, the genes come in different types, and all of us in this room are going to be of a certain type, either 129MM, 129VV, or 129MV. And it's just a normal variation. But if you're unlucky enough to get a prion disease, it may well affect how it affects you. Secondly, the structural form of the abnormal prion protein is key. And finally, there are almost certainly other factors in the person affected which we dimly understand, including perhaps other genetic factors that are nothing to do with the prion gene. So I mentioned lumping and splitting earlier on, and we started off with sort of CJD. We then sort of divided it up into sporadic, genetic, and acquired, and then we noticed that sporadic disease had differences in the clinical picture, pathological picture, and also differences in the diagnostic test results. And so that was started to be split up into subtypes. I'm not going to go into in great detail here, but they're basically divided into MM1, MM2, MV1, MV2, VV1 and VV2. I don't think you need to get bogged down in this detail. They're all forms of sporadic prion disease, and they're classified like this because there are differences in the clinical picture, the pathology and the molecular findings. I can't be giving the Pierluigi Gambetti lecture without mentioning variably protease-sensitive prionopathy, which, of course, he delineated first. But from my point of view, this is another form of sporadic prion disease, just with different rather characteristics, both at the clinical, pathological, and molecular level. But it all depends on whether you want to lump things together or whether you want to split them up.